Welcome back to another lecture. Today's lecture is about flower development in flowering. Okay, so when you look at a flower in a longitudinal section, like you see here, you see that there are four major organs. So those are organs that you are familiar with. They are the sepals, the outside leaves that cover the flower before the flower opens. The petal, those are usually the shiny colored ones. And then there is the carpel and the stamen. And so the carpel and the stamen, of course, carry the male and the female organs. Now, if you were to take a cross-section of this, then you get something that's called a diagram, a floral diagram. And that's often used by systematists who describe the flower in a shorthand. And so this is the shorthand for how many flowers, how many flower organs there are in this particular flower, such as the sepals, the petals, the stamens, and the carpels. And you can also see not only how many there are, so in this case 6, but also how they are arranged. So these are close together, these are further apart from each other, these two are close together. How many carpels there are, whether or not they're fused. So all of those kinds of things you can see, that is something that a systematist would like to know when they look at different types of flowers to see whether they belong to one species or another species. And then lastly, you can also draw this even more condensed in the schematic that just describes the whorls. So whorls are rings, essentially, rings of organs. And so there are 4 whorls in this particular flower, 4 rings of organs. And those then botanists have also described as fields, where they take 2 whorls that are adjacent to each other and consider them as fields. And you will see that these fields are actually quite important later on when we talk about the transcription of certain genes that determine whether or not you have flowers, whether or not you have stamens, carpels, petals, or sepals inside the flower. Okay, so but before we move on to those genes that determine how a flower looks, what a flower looks like, let's take a little detour here and look at some genes in flies. So there's a set of genes that are called homeotic genes, and homeotic genes are genes that control the development of body segments. So you'll later see that homeotic genes also control the development of body segments in plants and flowers, of course. But here, what I want to show you is some mutants that were found that determine the segment of this fly. So for example, here's a wild-type fly, and here you see a fly that has an extra set of wings. So this part of the body is essentially duplicated. And so a homeotic gene controls segments, and in a segmented animal, such as arthropods and flies, that can lead to the duplication of entire portions of the body. It can also lead to the placement of parts of the body in the wrong place. So this is a particular kind of mutation. We'll come back to that in a second, the type of mutation where a body part is put in the wrong place. But I wanted to show you this one, which is also controlled by a homeotic gene, which is called antennapedia, because it turns legs, or antenna into legs. So these are legs that are sprouting out of a place where there should be antenna. So this is a body segment that has been misplaced at the very frontal part of the animal here. So the definition of a homeotic gene is that homeotic genes encode transcription factors that are involved in specifying the location of organs. Okay. So let's take a look at homeotic genes in mice. There are some genes that are called Hox genes, and these Hox genes, when misexpressed, that leads to the formation of novel segmental structures. So here you can see that the spine is changed in the formation of these particular vertebrae here. So there's a close-up here of this part of the spine, where you see it's called ectopic arches. Ectopic means in the wrong place. So an arch, a vertebrae arch that is connecting certain parts of the body with each other that weren't there before. So this is what it should look like. This is what it looks like in this Hox mutant, which has ectopic expression of these genes and is making these vertebrae in the wrong place. So ectopic vertebrae. So mutations in homeotic genes often lead to ectopic expression of other homeotic genes. So those are the two terms for you to remember: homeotic genes and ectopic expression. So where is it expressed and what types of genes are these? So we'll see that also happens in plants. So if you look at number of mutants that were found that change the organ structure of flowers, you can see this list of 4 specific mutants, and you can also see how the flower has been structured in these particular mutants by looking at these cartoons. So in the wild type, this is of course Arabidopsis. You have 4 sepals, you have the 4 petals, you have the 6 stamens and the 2 fused carpels. And if you look at this mutant that's called apetalor, it doesn't have any petals, thus the name, but you see that it has additional carpels, right? There's a carpel here and a carpel here in addition to the carpel in the middle. And then it has the 6 stamen. So it has additional carpels, and they are of course ectopic because they're in the wrong place. They're in the outside whorl as opposed to in the inside whorl where they should be. And then we have a mutant called pistillata, and you can see that it has lots of carpels. The pistil here that has the carpels in which the carpels are fused together is multiplied, right? So you see all these extra carpels, and you see that there are sepals around it, but it's lacking the petals and it's lacking the stamens completely. And then this mutant here, that's called agamis because it's not making gametes, is comprised completely of sepals, petals, another whorl of petals, and then another whorl of sepals. And in some mutants, that's it. It just stops right there. But in other mutants, in other alleles of the same mutant, you get continuous formation of these whorls inside. So flower and flower, that's the agamis phenotype. So the mutants that I just told you about are the apetalar mutant, carpels, stamens, carpels, the pistillata mutant. There's also a mutant that's called apetalar 3, has the same phenotype, has only sepals and carpels. And then the agamis phenotype, sepals and petals only. Okay, so I'll make a little detour here. I wanted to remind you of a technique that we have briefly discussed before, and that was very important in understanding the function of the genes that encode these mutants that are responsible for these mutations I just showed you. So that technique is called in situ hybridization. And we discussed it before with respect to a way how you can measure the expression level of a certain gene. So to assess the gene, where a gene is expressed in the tissue, you can use techniques such as the fusion proteins that we've discussed before, the reporter proteins. But you can also use in situ hybridization. And so just in very briefly, what you do by doing in situ hybridization is that you take the tissue and you fix it. So in this cartoon here, it's just a single cell. But if you have to first section, for example, a flower, you would have to embed that flower into a wax block so that you can make thin sections. And then you take the fixed thin sections and you put them on a slide, on a microscope slide, and you produce a probe. And that probe is here shown with these little red dots, and those are labels. They can be fluorescent, they can be radioactive, they can be enzymatic, and then react with a substrate to produce a color. But they have to be complementary to the mRNA that you're interested in. So if you're looking for a specific location of a specific mRNA, and you're not making a fusion protein to see where it's expressed, you can use this DNA or RNA probe that you label, and then you hybridize it to your slide, you wash off all the unbound probe, and then you image it in the microscope, and you can see where these little dots are. That is where the mRNA is seen, and I guess the blue stuff is probably the nuclei. So this is a blow up of a certain section. So you have it as the probe, you view it under a microscope, can be fluorescent if your probe is fluorescent. So that is in situ hybridization. And now I'm going to show you a number of slides for all of these mutants that I just mentioned. And in those, you can see where that particular gene is expressed. So one thing very important to remember is that here we're looking at wild type, and over here we're looking at the mutant. So I just put the mutant here so that you see what the mutant phenotype is. But of course, in order to see where the RNA is normally expressed, you have to use the wild type. So in this case, the wild type was used. It was sectioned longitudinally. And so what you see here is an apical meristem. You just see the very tip of the flower, right? So this shows you an entire flower much later in development. This is just the flower as it's just starting. So we looked at those in the lab in longitudinal sections. This whole thing here is the floral apical meristem, the shoot apical meristem of the flower. And all the red dots are hybridization products of this particular probe for the gene A-pedalar 2 to the tissue. So what you can see is that it's expressed, that A-pedalar 2 in the wild type is expressed here in the outside worlds. The outside world also a little bit in the middle, but not in the inside worlds, right? So only on the outside. In this case here, you see where pistillata and A-pedalar 3 are expressed. So again, here is a section of a wild type, or this is just to show you where the different worlds are. And then here's a cross section of a wild type, and the signal here is white, so there's probably a false coloring afterwards, after the hybridization was done. But you can see one flower here. Here's another young flower that was right next to it. And you can see that the expression here is restricted to worlds 2 and 3. So it's not here on the outside, right? It's not here on the inside, but it's in these two adjacent worlds 2 and 3. And you can see that here as well, not here, but it is expressed over here. So that is where the genes A-pedalar 3 and pistillata are expressed. And then lastly, the agamis gene, again, this is the agamis mutant, this is an agamis wild type. Here you see one flower, here you see another flower, right? So here's the apical meristem, here's a floral bud that is sort of budding off, and here's the younger one, this is the slightly older one. And you see there's no expression on the outside worlds, there's only expression in the inside worlds. So put that all together, what I just showed you, and if we look at the expression patterns of these floral genes, we see that for AP2 and AP3 or pistillata or agamis, we have expression either in world 1 and 2, or in world 2 and 3, or in world 3 and 4. So researchers who were studying these floral mutants and who saw where these genes were expressed and who looked at the phenotypes and wondered what the genes might do, what their function might be in patterning the organs in the flowers, came up with this model that they called the ABC model. And so the ABC model tells you something about the expression, but it also tells you something about exclusion of expression of certain genes. So let's walk through this here step by step. So what this model suggests is that in these 4 worlds, 1, 2, 3, 4, there are 3 activity types, A, B, and C. And these activity types are correlated with the expression of the genes in these particular worlds. So we saw that A-pedalar 2 was expressed in world 1 and 2, where sepals and petals are produced. And A-pedalar 3 and pistillata, both of those genes were expressed in the B region, worlds 2 and 3, where petals and stamens are produced. And then agamis was producing stamens and carpels in worlds 3 and 4. And so the combination of expression of these ABC genes is what specifies which organ is produced. So what does that mean? It means that when you only have apetalar 2 being expressed, you will get sepals. If you're expressing apetalar 2 and apetalar 3 or pistillata at the same time, then you're making petals. But if this gene is expressed together with agamis, then that will produce stamens. And if agamis is expressed by itself, then it will produce carpels. The other thing that people noticed is that A and C class genes restrict each other's expression. So they cannot move into the other worlds. So oftentimes you see that sort of shown as these arrows that apetalar 2 and 3 are restricting each other, but when one of them is missing, then the other one can spread out into the other genes, into the other worlds. Okay, so let's go back and let's take a look at what that ABC model must mean for the mutants that we looked at before. So if we're looking here at an agamis mutant that has only sepals, petals, and then sepals again, the way we can explain that by the ABC model is that sepals are produced by an A-class gene, petals are produced by an A and a B class gene, then again in the next world, and then the absence of this C class gene that is restricting expression of A is no longer restricting the expression of A, and so now A can spread all the way out to all 4 of the worlds and can therefore produce sepals again where there is no B class gene to interact with. So this kind of expression pattern in the ABC model would explain this kind of mutant. And then likewise, if we have loss of A function, then we have the C-class gene spreading out into the first and second world. And that can now interact with the B-class gene in the second and the third world, producing only stamens there, while producing carpels in the outside of the first and the fourth worlds. And then lastly, where we have a loss of B-class function, this one is now gone. A and C are excluding each other. So they are both there, so they're excluding each other, and therefore they will only produce in the worlds 1 and 2 sepals, and in the worlds 3 and 4 carpels. And so with this model, people could explain the observations that they made from those floral mutants that they had isolated before. So it turns out that these genes from the ABC model belong to a class of genes that was already known. Well, some of these genes were already known, but together, some genes that were already known and these certain floral genes were categorized as a class of genes that are called MADS box genes. And so the MADS box stands for the beginning of individual genes. And so the apetalar is what gave the MADS box its A, and the deficiens is one, we'll talk about that in a second, is one that gave the MADS box its D. And then these 2 outside letters are from animals. So this is a kind of gene class that is found both in animals and in plants. And what all MADS box genes have to have in common, but here specifically it's shown for the Arabidopsis MADS box genes, is that they have an N terminal and a C terminal, like all proteins. And then they have this MADS box domain. A domain is simply just a region of conserved amino acids. So in that region, there's a number of conserved amino acids that you will find in all of these genes that are part of the MADS box class. And then there's a K box, same thing as the MADS box, but a different sequence, but it's also conserved. And then there's an intervening region or a variable region that's in between, that varies between the genes that are making up the MADS box class. In the MADS box itself, the domain that makes up the MADS box is a sequence of DNA that is known to have a function in DNA binding. So something these proteins that have this particular amino acid structure there have often been found in transcription factors that bind DNA, and that is the region of the protein that binds to the DNA. And the K box is a dimerization domain. So again, there the domain has been found often in proteins at that particular area where they bind to another protein. So this is a protein-protein interaction, while the MADS box is a protein-DNA interaction. Transcription factors binding to DNA, dimerization protein interaction, one protein binding to another protein. So with that, people already had a pretty good idea of what these proteins might be doing molecularly in the plant as they are specifying in the developing organ, in the developing flower, which organs were going to be produced. So again, the ABC genes specify the tissue development by either homodimerizing or by heterodimerizing, and that is where this K box comes in, that they can bind to each other. So an AP2 gene can homodimerize and produce sepals, or an A protein, of course. Agamous protein will produce carpels when it's homodimerizing, meaning two agamous proteins. But the heterodimers, where the B and the A class or the B and the C class are hybridizing with each other, binding with each other, that is a heterodimer. So two different proteins forming one complex. And so we have the AP2, AP3 that's producing the petals, or the AP3 and the agamous that's producing the stamens. So you can see that again here, right? So where you have the AP2, AP3, or pistillata, one of those two proteins, and then here you have the heterodimerization. Well, here you have homodimerization between two apetalate 2 or two agamous 2 proteins. So you might wonder, what does this look like if you have lost all of these genes? And that is really an interesting question. So when the first knockouts were made, where all of these ABC function genes were gone, people were surprised in some ways to see that a quadruple mutant, taking out the AP2, the AP3, the pistillata, and the agamous, gave a flower that was completely comprised out of vegetative leaves. So these are all leaves that sort of look like a flower. They're arranged like a flower, but they are green, they have chlorophyll in them. Other than where they are located, they are simply leaves. And so it's interesting in the way that long time ago, hundreds of years ago, philosophers of botany, people who were philosophers back then usually were rich people, and they had time to think about all kinds of things, not just about literature or the meaning of life, but they also looked at organisms. And so there was a well-known German poet, Goethe. He was more of a poet than he was a scientist, but he did do some science and philosophized a little bit about that. And he was the first who looked at all these flowers and said, well, it's really just a matter of leaves that are reorganized or restructured. So he had already in the 1750s or so proposed that flowers were really just differently developed leaves. As it turns out, that is exactly right. So a few years later, after the ABC model was understood and was proposed, as I showed it to you just before, additional genes were found, and these additional genes were then put into a called an E class. So the E class are these genes that are all redundantly required for the ABC genes to work. So it turns out that you need all of them in order for the ABC model to work. And so these E-class genes are expressed in all of these 4 worlds. If you have a mutation in all the E-class genes, so there are multiple E-class genes, and if you have a mutation in those, you get this phenotype that's called sepillata. So there it looks all just like sepals. So the E-class genes are required in addition to the correct ABC genes to specify flower organs. And like we discussed before, you can do you can do an overexpression analysis in addition to your mutant analysis to see, what happens, not only to see what happens when you knock out a gene, but also what happens when you overexpress that gene. And so overexpressing these E-class genes can turn vegetative leaves into floral organs, such as petals. So that was interesting and unexpected. Okay, so your book has this nice graphic where you can see all of these genes put together, where you can see the exclusionary properties of apetala and gametes, where you have the sepalata 1 through 4, and their requirement in all of the 4 worlds, the different types of organs that they're specifying. And then they also show you something that is now known as the quartet model of protein interactions. So that suggests that you need always 4 proteins, where we talked about the homo or heterodimerization, and that this quartet of proteins binds to the DNA in a certain way. So this is supposed to be the DNA that it's binding to. Remember that there were these boxes, right? So there were the protein-protein interaction boxes, but there were also the protein-DNA interaction boxes. And so the quartet model puts that all together to suggest how these proteins not only interact with each other, but also how they interact with the DNA. You can also see that on this slide here, where you have the MADS box binding to the DNA. So here's the DNA. So the CARC box is on the DNA. So that is a DNA sequence. The MADS box is the protein sequence. Then you have these intervening regions, the K region that is for the dimerization required, and then on the other end, you see that. So you see the dimerization here, and you see the MADS box on the other side of the DNA. So that gives you the quartet of these homeotic genes in their interaction, not only with other proteins, but also with the DNA. All right. In other species, this works as well. So it's not just a thing that you see in Arabidopsis, but you can see this in many, many other species. One species that has been studied quite well, but sort of been overshadowed more and more by Arabidopsis, but years ago was one model species that was preferred by geneticists. That is a snapdragon, or Antirrhinum. And in Antirrhinum, the same kinds of genes, the ABC genes that I explained to you and showed to you for Arabidopsis, have also been seen and characterized. And so an A-class mutant that has only carpal, stamen, carpal is called the squamosa gene. So unfortunately, they don't have the same gene names, right? They were isolated at different times, and then scientists gave them their names. And so they have different names in different species, even though they really have the same homologous function. So this is the squamosa gene that has the A-class function, producing carpal, stamen, carmen in the mutant phenotype. And then here's here 2 genes, just like we found in Arabidopsis for the B class. This one's called deficiens and globosa, and this is the D in MADS box, the deficiens, the D in MADS box. So this comes from the B class, producing sepals, carpal. So you see the sepals here and the carpals here. And then the one that's called plena, plena of course means full, is the C class, same as agames, and that's producing sepals, petals, and sepals. And it's a very full flower, very pretty horticultural variety of snapdragon. So horticulturists, of course, love these mutants, right? They have loved these mutants before the mutants were known as such, and before any of the molecular identity of this was known or function, and have developed flowers that have filled flowers, right? They're called filled flowers in horticultural lingo. So I have a picture here of some flowers that you can see. So this is a Brookmansia. Brookmansia are these trumpet flowers, I think they're called also. So here you have a wild-type flower, and then here you see some extra petals, and here you see a mutant that has a flower within a flower. So this is sepals, petals, sepals here, the sepals, maybe sepals inside, maybe the sepals on, here you see some sepals. So they're a little bit changed here in appearance as well. And then here's an example of a replacement of all the inner organs, the carpels and the stamens, and you have the formation only of petals. In the very so there should be some sepals in there, but these sepals seem to turn color into pink, and so this variety is called pink perfection. So this is another horticultural variety. And if you look in a garden center, or you ask a horticulturalist about filled flowers, you will see that many varieties exist that have extra petals, and that in many cases, of course, it's not known for all of them unless people have taken the time to study to see whether that is the kind of gene expression that is changed in that particular species. But in many cases, it seems that that is the result of ectopic expression of these homeotic genes in the flowers, making them so pretty. Or maybe you like this one better. Regardless, that is how homeotic flowers work, and that is the end of today's lecture. See you in class.